Okay, so dear all, welcome. So it's 5 p.m. We are ready to start. Welcome again. I'm Mariangela Pellegrini. I work for the European Reference Network on Rare Hematological Disease. I'm very honored and happy to welcome indeed all of you to this first webinar. So this webinar is indeed a joint project of the EDIT Consortium and the ERN-EuroBloodNet. So we have designed 7 webinars with a focus on genetic therapy for people living with sickle cell disease. And so we will provide indeed an overview of gene therapy and genome editing approaches for sickle. We are very proud of this project because it has been, as said, developed in partnership with the Sickle Cell Disease Project, who is led by Annarita Miccio from Imagine Institute of Genetic Disease in Paris. But we have also benefit from the contribution of a steering committee and mostly of patient representatives that I really want to thank today. So Miriam Santos Freire, Dianaba, and David Issom, thank you so much for helping us in creating a patient-centered project. So before starting, next slide, please. Some housekeeping rules. Thank you very much for listening. So first of all, as you can see, your microphone is muted and will remain muted during the presentation. As you can notice, this webinar is recorded because all the videos will be implemented in both e-learning environment of the ERN and EDIT Sickle Cell Disease webpage. So you will have access to all the educational resources. And you can write your question in the chat at any time. We will take the question during the Q&A at the end. Last slide. We have a new tool for artificial intelligence. How to use it, please follow the screenshot here. You can see, so you have this symbol like choose C at the bottom of the bar in Zoom. You can click on it and then you will select the translation in the language people are speaking. So my speaking language, we are speaking English, and then the language you want to have the caption. So if you want to use this tool, you know how to do it. If you have any question about this tool, if you are struggling, please write in the chat. We will guide you through the tool. And so next slide, please. And so, yes, so today is the first webinar. We will talk about introduction to sickle cell disease and gene therapy. With us, we have 2 speakers, so Annarita Miccio from Imagine Institute and Professor Michaela Semeraro from INSERM and AP-HP.Centre Université Paris-Cité. So I will pass the floor to Annarita, who is also the coordinator of sickle cell disease. So thank you very much, Annarita. The floor is yours.
Thank you very much. Can you see the screen?
Yes, we can see and hear you too.
Okay. So good afternoon, everybody, and welcome to everybody. So my name is Annarita Miccio, and I'm directing a research lab at the Imagine Institute in the Necker Children's Hospital campus. And our goal is to develop a gene therapy strategy for sickle cell disease. And so today with Professor Michaela Semeraro, who is also working at the Necker Hospital and is taking care of innovative therapy for patients with genetic disease, such as patient indeed with sickle cell disease. What we will do is to start the first of a series of webinars that are focused on gene therapy for sickle cell disease. And so, as Mariangela said, this was a joint effort in the frame. And so the webinar were organized in the frame of the EU-funded EDIT Sickle Cell Disease Consortium in strict collaboration with the ERN-EuroBloodNet, and the design was done also in collaboration with the patient, and this was extremely useful. So we don't have any conflict of interest to disclose. And so what we will talk about today will be, first of all, and we will give Dr. Semeraro will give an overview on the history of sickle cell disease and the current distribution worldwide. And then we will try to understand what causes sickle cell disease. So what is the hemoglobin and how mutation that affect the hemoglobin genes can cause sickle cell disease. And then Dr. Semeraro will give a brief overview on the current therapies for sickle cell disease that you probably know very well, with all the advantages and drawbacks. And finally, we will introduce the genetic therapies and in particular gene therapy approaches for sickle cell disease. And this is, again, the first webinar, so it will be a general introduction of the sickle cell disease and gene therapy. But then at the end of this webinar, we will also introduce the other 6 webinar in which we will detail much more this gene therapy strategy. And so for now, I will leave the stage to Professor Semeraro.
Thank you very much. It's a good afternoon, everybody. I'm very happy to be with you today. I would like to start with a brief reminder of the history of sickle cell disease. The next slide, please. Because as you know, the sickle cell disease is the first genetic disease studied by science and is also the first disease with an approved gene editing therapy. And we will see this later, but just to remind you that the first clinical description of sickle cell disease was performed by an American doctor, James Herrick, in 1910. And then the molecular structure of the sickle cell disease was described by Dr. Pauling in 1949. Then with the sequencing of the DNA, the hemoglobin chain were the first described and then allowed the diagnosis, the molecular diagnosis of sickle cell diseases. And then with these capacities, we were able to test patients in order to detect sickle cell diseases as a newborn screening for patients, and the newborn screening becomes widespread and is actually a reality in several countries. And then the last step that what we will talk together today will be the innovative treatment with gene editing, with the first CRISPR-based therapy with the Casgevy, which has been approved in UK and the US in 2023. The next slide, please. Sickle cell disease is not a rare disease since it affects more than 8 million people worldwide. And moreover, in Europe, there are 52,000 people with these diseases and 100,000 people in the United States. Most of the patients are of African origins, and but with the migration, the sickle cell diseases is spread far beyond Africa, and today can be found all over the world. So the next, please. Why why this origin in Africa, in the Middle East, and in Asia? Because this is for a history reason, because the sickle cell disease trait was able to protect against malaria, which is an infectious disease with the endemic in this specific region. And this is the reason why over time this trait became frequent in this population. But because of the impact, and no, you can stay already in the previous slide, but because of the impact of the sickle cell disease all over the world, Europe recognized the sickle cell disease as a public health problem in 2008, and then several countries of the European Union recognized the importance of working on strategies to improve the diagnosis, the treatment, and the care of patients with sickle cell disease. In order to explain you in the next slide what is it, the sickle cell disease, let me explain some information about the molecular basis of sickle cell disease. And first of all, we have to talk about hemoglobin. And just to remind you, the next slide, hemoglobin is a protein present in our red blood cells, so in our vessels, and the objective of the aim of the hemoglobin is to carry the oxygen from the lung into the rest of the bodies. Moreover, hemoglobin interacts with the other gases, and the protein of the hemoglobin is made by 4-chain for protein, but these proteins are not always the same during life, since in the prenatal period, the hemoglobin is able to change with the different chains forming the hemoglobin protein. And for instance, there is an embryonal hemoglobin and a fetal hemoglobin. And this is very important. You will see for the target therapy that we will approach later, because at the fetal stage, we observe a fetal hemoglobin with 2 alpha chain and 2 gamma chain. And this hemoglobin is characteristic because the affinity for the oxygen is more important, because it's more important during the pregnancy period. And while during the adulthood, the hemoglobin switch with an adult form with 2 alpha chain and 2 beta chain. It's important to note that there are some paraphysiological anomalies in some patients, and for some patients with a specific disease named HbFH, there is a persistent form of fetal hemoglobin, and normally this condition is harmless, so it's a benign genetic mutation. And the next slide, please. So what is it at the molecular level, the sickle cell disease? There is a typo in the DNA. This typo occurs in the DNA coding for the beta globin gene, which is located on chromosome 11. And this modification is able to induce an abnormalities in the fiber constituting the red blood cells, and so the red blood cells are not round as in healthy people, but they have a specific shape, a sickle shape. So in the next slide, we say that sickle cell disease is a genetic disease with an inheritance of sickle cell disease, and the type of the inheritance is an autosomal recessive condition. What does it mean? It means that we can receive, as for all the people, we receive our gene from our parents. So we receive 1 copy of the gene from the father and 1 copy of the gene from the mother. So if the parents are healthy carriers, which means that they represent just 1 copy of the gene modified, the probability to have an affected child is 25% of chance, while the possibility of having an unaffected child, so without sickle cell disease, is also 25%. 50% of chance for children with the healthy careers like the parents, which mean with just 1 copy of the sickle cell gene. And in this case, there is 1 gene mutated, so there are no symptoms associated of the diseases. The next slide, please. You already know which are the symptoms of sickle cell diseases because this typical shape of the red blood cells induce a chronic hemolysis, chronic hemolysis is a quantitative anemia. So the hemoglobin is not high, and so the red blood cells break in the vessel, and this typical shape of the red blood cells and the chronic hemolysis induce a vascular occlusion in the small, especially in the small vessels. And this vascular occlusion is responsible of the symptomatology with the pain, with the vaso-occlusive crisis. And moreover, when the vascular occlusion occurs in small vessels, in specific organs like central nervous system, lung, or kidney, there are some, there is a risk, an increased risk of life-threatening. So the next one. And so what we can do to treat sickle cell disease, I would like to perform an overview of the current therapies. I will talk about the more common therapies, but the research and the trials ongoing are different, and there are other different kind of approach to treat the crisis, but also basically the disease itself. First of all, we will talk about hydroxyurea. Hydroxyurea is a drug, is an oral drug. Normally, it was developed as a chemotherapeutic drug, and in the specific of sickle cell disease, this is very important because this drug is able to increase the fetal hemoglobin. You know, it's the fetal hemoglobin present during the prenatal period. And in increasing the fetal hemoglobin, there is a balance with an increased fetal hemoglobin, and so a consequent decreasing of the sickle cell. The hydroxyurea are indicated so in patients in order to reduce the sickling and to reduce the related complication. At the end, the aim of giving this drug is to improve quality of life and survival. It's very easy because to take, because an oral drug, you have to take this drug each day, but there are some risks associated, which are, since this is a chemotherapy, there is a risk of low blood counts, and so an increased the infection could be increased. It could be not very well tolerated from a gastrointestinal point of view. It can induce skin or nail changes and possible fertility problems. The next one, please. Second kind of treatment are the red cell blood transfusion. Red cell blood transfusion from healthy donors. The objective is to substitute the sickle cell with normal red blood cells. The transfusion can be sample or can be the exchange. The two differences, the differences between these two kind of transfusion are that the simple transfusion add healthy red blood cells without removing the sickle cell, and it's indicated before surgery, during pregnancy, or if there is a severe anemia with a chronic hemolysis. And while the exchange transfusion improve and reduce, remove actually the sickle cell already present in the patients and substitute this sickle cell with the red blood cells of the healthy donor. And the exchange are more indicated in stroke prevention, in acute chest syndrome treatment, or in silent or not silent strokes. But this transfusion have some risks, because the risks are linked to the transfusion itself. So an immunization, the iron overload, because all these red cells given to the patients can break, and then there is the iron inside the red blood cells that could be delivered into the body. And other risks are the transfusion, the reaction, the infection, but these are rare because now are very well controlled. And for sure, for the patients is could be very complicated to receive each month or each 2 or 3 months, according to the calendar of the red blood cell transfusion, the red blood cells, and even because it's difficult to have a venous access for patients who receive regularly blood cell transfusion. So the next one. At the end, so the only effective treatment for sickle cell disease approved is the hematopoietic stem cell transplantation. What does it mean? The hematopoietic stem cell transplantation, it is a substitution of the stem cell which are in the bone marrow of everybody with the new stem cell derived from a healthy donor. This is, it works like a transfusion, but in order to have a space inside the bone marrow to receive the new bone marrow cells from donor, the patients needs to receive a conditioning regimen, which is a high dose of chemotherapy, and then the cells can be transfused, and then there is a period in order to allow the growing and the multiplication of these stem cells, and at the end, the patients can, the stem cells given to the patient are able to produce healthy red blood cells. This treatment is, it works, but there are some risks linked to the transplantation itself, with a risk of disease relapse if it doesn't work, of organ toxicities, infection, cardiovascular disorders, and rejection of the cells with which is called graft versus host disease. Moreover, in order to receive a hematopoietic stem cell transplantation, you have to be the donor has to be very similar from a DNA point of view to the receiver, because there is a risk of intolerance between the two between the cells. So most of time the donor is a sibling or a donor from a registry with the specific characteristics, specific genetic characteristics in order to receive the transplantation. The next slides. So this is an overview of the treatment and of the sickle cell disease. But if you would like to explore these points in more details, I invite you to view this video and these websites in order to go through with these links. So now I will let Annarita talking about genetic therapies for sickle cell disease.
Thank you very much, Michaela. So basically, just to summarize, so far we have discussed about therapy that are addressing the symptoms of sickle cell disease, or we have talked about bone marrow or hematopoietic stem cell transplantation, which is a curative therapy and is a one and done therapy, but this is available only to a fraction of the patient, as Dr. Semeraro explained, so only the patient that have a compatible donor. And therefore, here we will discuss instead the gene therapy. And so the concept of gene therapy is really to provide a therapy to potentially all the patient because we are genetically modifying the cells that come from the patient. So we don't need a donor that could give us his good hematopoietic stem cells. And in particular, gene therapy is a technology that really aims to treat genetic disease by addressing their root cause. Just to give you an example, we want to address with this therapy, for example, we can address the mutation that is causing the sickle cell disease, so the typos that cause the production of this abnormal hemoglobin. So in particular, the scientists have followed two different approaches that both have reached the clinics. So the first is called a gene addition approach, and the second is a gene editing approach. So in the first case, the gene addition approach is based on the insertion of a normal copy of the gene into the cells of the patient without getting rid of the mutated gene. So the two genes coexist, but the normal gene can produce the normal hemoglobin in the case of gene therapy for sickle cell disease. And usually we deliver this normal copy of the gene with a viral vector. And in particular, this type of strategy, this gene addition gene therapy strategy has been used for many diseases and has been successfully used for many diseases such as retinal disorders, hemophilia, and of course also sickle cell disease. And in particular, in sickle cell disease, we use a vector, a viral vector that we call lentiviral vector that is able to permanently integrate this normal copy of the gene into the genome of the patient. And since this is a permanent integration, this means that this therapy will last forever in the patient. And in parallel, gene editing approach have been developed. And in this case, gene editing is a technique that aims directly at changing the patient DNA. And therefore, what we can do in this case, if this is the mutated gene, again, the beta globin gene in the case of the sickle cell disease, what we can do is to use these gene editing tools that we also call molecular scissors, basically to correct the mutated gene so that we can restore the normal gene and as a consequence, the normal hemoglobin production. So basically, but how does it work? So the procedure of gene therapy is really quite similar to the procedure of the bone marrow transplantation, but of course we are using and correcting the patient cells. So in particular, we collect the cells from the patient, and then the cells are genetically modified ex vivo, which means that we take them from the patient and we culture them in the lab. So these are genetically modified either using a gene addition approach, in which, as I told you before, we use a viral vector that brings a good copy of the gene, of the hemoglobin gene in our case, into the patient cells, or we use gene editing approaches. So gene editing approach, as I mentioned to you before, can correct the mutation in the beta globin hemoglobin gene that causes the sickle cell disease, or as we'll see later, these gene editing tools can also be used to activate some genes, such as the fetal hemoglobin gene, and so to increase the level of fetal hemoglobin that, as Dr. Semeraro explained, are very beneficial for patients with sickle cell disease. Then these cells are then reinjected into the patient, and in the meantime, the patient has received a chemotherapy to get rid of all the mutant hematopoietic stem cells, possibly, and also to make space in the bone marrow for the newly genetically modified hematopoietic stem cells. And then these hematopoietic stem cells in the patient will then engraft and hopefully give rise to normal red blood cells. This as final aim will be then to improve the symptom of the sickle cell disease, so reduce the hemolysis, the death of red blood cells, increase the hemoglobin concentration, and reduce the vaso-occlusive crisis and therefore the pain crisis. And this therapy has been already successfully used in several trials and in several patients. And again, we are still modifying the DNA. So this is a one and done therapeutic approach. Despite, of course, the potential risks that are associated, for example, to the entire procedure, including, for example, to the chemotherapy regimen that is still required in these approaches. Okay, so where are we for the gene addition approaches? So there has been a significant progress in gene addition therapies in the last 10 years, and scientists have obtained functional cure of several patients in multiple clinical trials, but the most common and the most known therapy is the lovo-cell therapy that was developed by the company called Bluebird Bio, and this is a therapy, again, based on the use of a lentiviral vector that can carry and deliver a normal beta globin gene to the hematopoietic stem cells of the patient that then will give rise to normal red blood cells and not anymore to the sickle cells. So this therapy has been approved in 2023 with the brand name of Lyfgenia by the FDA, and indeed most of the patients that have been treated with this therapy are free of vaso-occlusion crisis, and many of them also are free of red blood cell transfusion, and thus representing an important breakthrough in the field of gene therapy of sickle cell disease. I just want to notice that this therapy has been approved by the FDA, but not in Europe. So this is available only in US at the moment. And of course, to know much better about this therapy and the clinical trial associated to this type of approach, we will have a second section, a second webinar on the gene therapy with the lentiviral vector. And then the second type of approach that scientists have undertaken is a gene editing approach. So gene editing is basically a technique that allows to change DNA. And so if we can change the DNA, of course, the ideal strategy would be really to change the mutation and so correct the genetic mutation causing sickle cell disease by reverting the mutation. And in particular, to do so, we use the CRISPR-Cas9 system, which is this represented as molecular scissors that can indeed cut a specific region of the genome, in particular, in our case, the beta globin gene. And in particular, while the cells tend to repair this cut in the mutant gene, at the same time, the cells is able also to correct the mutation and so to achieve the restoration of the normal gene function. We call this system, we call it like a double scissors, because as you may know, the DNA is composed of two DNA helices, and this system is able to cut both of them. Then this system has been evolved, so starting from the CRISPR-Cas9, the scientists have developed more precise and potentially safer tools, such as the base editing and the prime editing. And as you can see, the base editing or the prime editing, to be active and to be functional into the cells, they only need to cut one of the two helices of the DNA, and thus for this reason they're considered to be safer. And just briefly, but this will be discussed in the next webinars, base editor can correct only some type of mutation, but actually it's very, very efficient. Indeed, as I will show you later, it is also already been used in clinical trial. While prime editing regarding the editing efficiency is still need to be ameliorated, but is able to correct all the different types of mutation. Oh, sorry. So how we can use gene editing to treat sickle cell disease? So we can treat the patient hematopoietic stem cells with the CRISPR-Cas9 base or prime editing system, and the goal is to correct the mutation or to activate the fetal hemoglobin gene. So in the first case, if this is the mutated gene, this is the typos in the beta globin gene that cause the formation of the sickle cell disease or HBS. In this case, this sickle hemoglobin determine the formation of these deformed and sickle-shaped red blood cells, while with the gene editing, we can correct this typo in the beta globin gene, thus leading to the generation of a normal hemoglobin, which is the HbA, and this one can then allow the generation of normal red blood cells. A second strategy aimed to reactivate fetal hemoglobin. Indeed, the elevated level of fetal hemoglobin are beneficial for sickle cell disease patients. Indeed, the mechanism of action of hydroxyurea is really this one, to increase the level of fetal hemoglobin, that is a good hemoglobin for the patient. But normally in adult cells, the gene that are producing the fetal hemoglobin are inactive, so we don't have fetal hemoglobin, and so the patient have these deformed sickle red blood cells. So in this case, the gene editing strategy aims at activating the gene that encodes for this fetal hemoglobin, thus inducing high level of fetal hemoglobin, and thus leading to the generation of normal red blood cells. And indeed, where are we at for the gene editing, especially regarding the clinics? Actually, really the editing strategy that aim at increasing the fetal hemoglobin level, those are the strategies that have been currently have been successfully used in clinics. And in particular, you might know about Casgevy, which was the first gene editing strategy for sickle cell disease that was approved in 2023 by several agencies, the UK, but also the EMA and the FDA agency. And this is a strategy that is based on gene editing, in particular on this CRISPR-Cas9 system, again with the goal to increase fetal hemoglobin level. And this strategy has been very successful, with most of the patients being free of vaso-occlusive crisis and also free of red blood cell transfusion. Following this trial, another company, Beam Therapeutics, have developed the Beam 101 product that has the same goal to increase fetal hemoglobin, but this time the scientists have used the base editor. So this therapy is currently being tested in an early clinical trial. So we call them phase 1, phase 2, because they are really just the first trial where this therapy has been tested to evaluate mainly the safety, but also then the efficacy of this treatment. And the early clinical data of this trial that is taking place only in US showed very promising result with very high hemoglobin level and no vaso-occlusive crisis. Of course, these two approaches will be discussed much more in detail in the session 3 and in the session 4, so in the next webinar. And finally, I would like to invite you to listen to the story of Jimmy and Victoria to understand the potential really of this gene editing strategy. And in particular, Victoria and Jimmy are amongst the first patients that have been treated with the Casgevy therapy. Victoria is a mother of 4 from Mississippi, and she was really the first person that was treated with the Casgevy in 2019. And after she was treated, she did not have any more pain crisis, or she was not anymore dependent on red blood cell transfusion, and she was also able to stop pain medication. And she's now really living a happy life with her family, working and traveling. And those when she tells this story, you will hear that these are things that she would have never thought to be possible for her. Another interesting story is the story of Jimmy Olaguerre, who is a tech entrepreneur from Atlanta. So he was also amongst the first men treated with the Casgevy therapy in 2019. And after a few months, he tells in his story, he tells that all his symptoms have disappeared. And incredibly, actually, in 2024, he climbed the Mount Kilimanjaro, and he was the first patient with sickle cell disease that was able to do it with the aim to bring awareness to sickle cell disease, the Casgevy therapy, and also to its accessibility. And when he tells his story, he really says something like, after the treatment, it was like if I was reborn. So I really invite you to follow this very interesting story. So finally, I would like to introduce the next webinar. So this was a general and not so much detailed webinar, but we will go more in detail in the next one, in particular in the session 2 to be held in November 2025 with Professor Giuliana Ferrari. We will discuss the gene addition approach with the lentiviral vector that are in clinics. And also in session 3, we will discuss with Dr. Mussolino and Dr. Amendola in February 2026 the CRISPR-Cas9 approach with the Casgevy trial and clinical therapy. And then in session 4 with Marcello Maresca, we will discuss the novel editing tools such as base and prime editing that have been derived and optimized starting from the original CRISPR-Cas9 system. Importantly, in session 5, we will really address the safety of the CRISPR-Cas9 system and its evolved derivatives with Dr. Ayal Endel and Professor Tony Katomen. This will be in May 2026. And in session 6, I will discuss the future development of this therapy to reduce the cost and improve the accessibility. And finally, in session 7, which will be also very interesting with David Morrow from EATRIS, we will discuss the regulatory path to the clinic. And finally, I would like to acknowledge all the members of the Dr. Michaela Semeraro and all the members of the EDIT Sickle Cell Disease Consortium that will contribute to these webinars, and of course the ERN-EuroBloodNet, and in particular Mariangela, together with all the patients for really the design, the organization, and the discussion about this webinar, and also the medical writers, Martina, Antonio, and Malel. And finally, I would like to thank all of you for your attention, and we will be happy to share, to answer to your question. Thank you.
Thank you very much, Annarita and Michaela, for your comprehensive presentation. So now there's the possibility to ask questions. So don't hesitate to write in the chat. Indeed, I'm seeing that the first one has arrived. So I will read it. Can the patients develop blood cancer after getting a gene therapy?
So maybe I can start, then Michaela can follow me. So basically what the gene therapy is not without risk. That's something that we always say. What I can say is that regarding especially the blood cancer, I can give you our experience here at the Necker. So the first patient that have been treated with gene therapy were treated with first generation viral vector that we do not use anymore, and especially we do not use for sickle cell disease. And so these were patients that Dr. Semeraro actually is still following after 20 years. And so basically there were with this first generation viral vector, there was, it's true that there was the risk, we discovered after that there was a risk of developing blood cancer. And that's why for years we have worked a lot to improve the safety of this viral vector. And I have to say that in the case of the sickle cell disease, with over 200 patients treated worldwide with lentiviral vector, there have not been any cancer associated to the lentiviral vector used for sickle cell disease. And this is for, let's say, the gene addition approach. For the gene editing approach, for sure, is much more recent, but we have learned a lot from the lentiviral vector study. And again, so far today, I think around 100 patients, even more, have been treated. And as far as we know, but data has been published, this gene editing strategy is very targeted. We are just modifying the globin genes. And so far, there are no really no report of blood cancer due to that, but this is something that we monitor constantly. Maybe Michaela can add some.
Yeah. Actually, yes, with the current gene therapies, we don't observe the risks observed in 20 years ago with the first patients treated by lentiviral gene therapy. So for sickle cell diseases, gene addition and gene editing are demonstrated good safety profiles, but for sure we have to improve our data with long-term data, and we need to follow the patients for several years in order to be sure that the safety is constant over the time.
Maybe if I can add something, in particular for sickle cell disease, we are particularly attentive on this point because still the population with sickle cell disease might have a higher incidence on some type of cancers, but actually we check also when we select the patient, there is still a phase of selection of the patient. So basically there is, if there is any doubt that this patient can be predisposed to have a cancer, of course we don't treat the patient because we don't want to give another hit that can stimulate something. This is, we really do a huge screening, a big screening to be sure that the patient is, let's say, in the better shape, in best shape it can be.
Thank you. So we go with the next question that is about eligible criteria. So thank you for this great presentation. Which patients can access the gene editing approaches? For instance, severe, moderate patients.
So I can reply because thank you very much. It's a very good question, even because it depends on the countries. So where there is an access for the gene editing and the gene therapy treatment, the criteria to benefit of this treatment depends on the country. Most of the European countries allowed this treatment just for severe cases. And as Annarita said before, the clinical characteristics of the patients are also important because there is the presence of some concomitant disease could be an exclusion criteria, and each eligibility to the gene therapy needs to be discussed by a consortium.
Thank you so much. So I will follow with the same topics, the following two questions. Are there any criteria for choosing candidates? Well, it depends on the country. And is this therapy possible in children?
Yes, for sure. Actually, we prefer to treat children because, as I told you before, the concomitant diseases occur normally later in life, and so treating children is easier, we can say, than an adult one. But normally the authorities in different countries, and I can say that in Europe is the case, prefer to treat with the innovative treatments at the beginning the adult population and then the children, because the follow-up of the patient, the safety is easier to follow in adult patients compared to the children.
So for example, if I can add something, the Lyfgenia therapy, for example, it has been approved for children older than 12, while the Casgevy therapy, let's say, is more open, and now it's approved drug. So it's also up to the doctors to choose the candidate. But actually, I think there's still discussion because Casgevy has been approved, but still there are discussion with the reimbursement agency in each European country, at least, and everywhere in the world. So still the criteria might be sometimes might be also a bit strict. Still, although it's open to all of them, but what they suggest or what they discuss is to have to treat patients between 12 and 35. Again, because they feel more comfortable of treating patients that are older than 12, while after the 35, there are a lot of comorbidities, and so this is this could impact could impact really the success of the therapy.
Thank you so much. I will take the next one. What is the total time gene editing take for patients from diagnosis till treatment? So it could mean two different questions. So the age, what we were saying, and then the time for the treatment itself.
Annarita, if you agree, I can start. There are different scenarios, because if the patient is addressed directly to a gene therapy, the treatment could be like a bone marrow transplantation, because you have to consider the collection of the blood stem cells, and then the hospitalization with the conditioning regimen, and then a period of aplasia in order to allow to the blood cells to grow up. And if you consider the collection and the treatment, at least 2 months of treatment, but sometimes could be more.
So if I can add something for the diagnosis, I think this patient, at least in Europe, they are diagnosed very early, because the symptoms around 2 years old, they already have symptoms even before some time.
And there is a screening also.
There is a screening. Depending on the country, we have some.
Yeah.
And so this is regarding the length of the treatment, what we will discuss maybe in the last webinar are the new development, because still now, for example, with the current therapy, it takes a while for the hematopoietic stem cells to produce all the different blood lineages, because still we are treating the hematopoietic stem cell outside of the body. So this is not something that is physiological. So there are novel therapies that have been developed, and even in the last trials, there's a time in which the hematopoietic stem cells give rise to all the blood cells and repopulate the individual with sickle cell disease is getting shorter, because we are improving the way in which we treat and we correct the cells. But this is a good point, because I think that this we can reduce the length that could be sometimes longer, up to 6 months from the recruitment of the patient to the treatment.
Thank you so much. The next one. Is this eventuality possible for patients with cerebral vasculopathy?
So unfortunately, in the current trials, this is an exclusion criteria. But we hope that with the proof of the efficacy of this treatment in the next trials and in the next treatment, we will able to treat also patients with this concomitant disease, with this consequence of sickle cell disease.
I think the criteria indeed, it's true that we treat severe patients, but not those for now with cerebral vasculopathy. But severe patient, maybe Michaela, you can comment on it. I mean, still the criteria to define a severe patient
Yes.
also vary from country to country.
Yeah.
Maybe Michaela, you can say what we.
Yeah, you're right. The severity is linked to the number of vaso-occlusive crises and acute chest syndromes. But the exclusion criteria are the presence of stroke, of cerebral stroke, because of the complication linked to the condition and regimen.
Okay. Thank you. Okay, so we go with the next one. What's the efficiency of gene editing in HSC with prime base editing?
Yeah. So this is a very important question because I think with the genome editing we are improving the precision, we are improving safety, but the efficiency is an important criteria because while for some diseases such as immunodeficiencies we might need to correct 5–10% of all the cells, and this is enough. In the case of sickle cell disease, I think we need to correct more than 50% of the cells probably to have a complete correction of the phenotype. And so for prime and base for base editing, it has become really very high. So in the clinical trial with bean therapeutics, is 90% of the cells after 6 months are already all genetically modified, which is great because these translate in a very good clinical data. While with the prime editing, I have to say that it might work even better biologically because we can really do some fine modification. But so far, I think that the highest, for the sickle cell disease, the highest efficiency that we and others reach are around 30%, which is something that we should ameliorate if we want to go in clinics, because we know that this might not be enough. And also because when we have 30% of the cells that have been modified, they've been treated, they received all the gene editing tools, so they've been, let's say, we try to treat them well, but still they receive something inside. And while the 70% of the cells, they do not, so they might be healthier or more fit in repopulating the body of a patient, which means that if we inject only 30% of edited cells, probably in the patient, only 10% will survive. So we need to go probably above 50%.
Thank you. About the cerebral vasculopathy we were naming before, one person in the audience is asking, have you done any studies on the genes involved in patients with cerebral vasculopathy? If so, can they be shared?
I think there have been many studies in the sickle cell disease population regarding the association of the other gene, because it's true that the sickle cell disease is extremely variable. There are patients that have some symptoms, patients that do not have. So there are many geneticists that are studying which are the genes that could contribute to this worse phenotype or to a better phenotype. And actually, I think that so far the studies are very sparse, which means that are done in very different centers with the limited number of patients. But now and so, I mean, we read many of these patients because we had questioned many of this paper, but none of them was really giving you a clear answer. But the good thing is that nowadays, for example, in US or in UK, there are large studies. In US, they are studying 80,000 individuals with sickle cell disease, really to discover this association between genes, particular variant of genes, and these ACS or these different symptoms. So I might advise you some papers, but I'm not sure those are really the genes that will be.
Yeah. If I can add something, actually there are several genes that are involved in cerebral vasculopathies, and so the aim is to explore if there are some association of known genes, like in Moya disease or in APOL1 kidney disease, with sickle cell patients. Because one patient can bring both the gene modification for sickle cell and also for endothelial cells or other genes involved in vasculopathies.
Thank you. So I think we have one last two questions. Did you encounter off-target effect while using CRISPR in your work in ex vivo?
Yes, so I can answer. So basically, indeed, you can have some. The idea, the beauty of CRISPR is that theoretically, they have we will discuss in the next webinar a GPS that really can bring the system only to a specific locus, for example, the beta globin gene. But this might not be always the case. So we are doing an extensive analysis of the off targets, which means if with this molecular scissor we cut somewhere else in the genome, we really do not want it. But we have some flexibility, so we can choose the strategy that has less off target. And if in any case you find an off target, what we can do in the lab is to study the effect of this off target, because maybe you are making a change in another gene or in another region which does not have any effect. And so in this case, it's also a balance between what you want to achieve, the cure that you want to achieve and the risk. But if we demonstrate that this change that we are doing in an off-target locus in the genome is not does not have functional consequences, I think this is enough to be able to then bring this cure to the therapy, this strategy to the therapy, to the clinics, sorry.
Thank you very much. We take all the questions. That's been so interesting and clear. Thank you so much. So before closing and thanking the audience and the speakers, I would like, yes, thank you, Gavin, to tell you that if you want to register to the next webinar, we have just put the link in the chat, and we also have QR code if you want to register with your mobile phone. And the next one will be in 2 precise months, meaning the 26th of November. We will have a trio of experts, so Annarita Miccio, Michaela Semeraro, you know them already, and Giuliana Ferrari from Ospedale San Raffaele in Italy. And we will talk about basics of gene therapy with lentiviral vectors. So we will delve more into the technicity of those therapies. And so thank you very much. It has been really interesting. Thank you so much, Annarita and Michaela, for sharing with us your expertise and time. Really, it has been excellent. Thank you so much to all of you. And just for information, we will publish the video of these webinars soon. So if you want to review it before coming to the next one, it will be soon available. So thank you very much.
Thank you. Thank you.
Thank you.
Thank you.
Thank you for the participation.